In this video, we need to look at what happens prior to release of neurotransmitters. So how exactly do the neurotransmitters get released? How does the action potential make that happen? There are pieces I haven't showed you yet, and so I want to show that to you. So this diagram shows a lot of what's going on. So the action potential, if you go back and review what an action potential is, it travels along the length of an axon, and then this is the axon terminal, or the terminal bouton. And so what is actually moving? Well, why don't you pause the video and take a minute to just walk yourself through what is moving in an action potential? What do you see move following these arrows? I hope what you talked about was the change in membrane potential, because that's what's happening. What moves along here is a region of very positive membrane potential, positive 30 millivolts instead of the regular negative 70. And when it moves past, so right here where it's red, this would be positive 30, and then back here behind it where it just was, just a moment ago, it's going to hyperpolarization towards minus 90. And in front of it, it hasn't gone through the action potential yet, so it's minus 70. So that area of very positive membrane potential moves. And what that means is that there can be other voltage-gated channels that respond to that. And there are voltage-gated calcium channels. And you'll see this a lot as you go forward in A&P. Calcium moves in, and calcium does a lot of things inside the cell. And so what calcium is going to do is cause an interaction between what we call snare proteins. I don't remember or care what it stands for, but vesicles have what's called V snares or vesicle snare proteins, and the membrane has target snares or T snares. And when these two proteins interact with each other, it will pull the vesicle into contact with the cell membrane. And when it fuses, whatever's inside the vesicle will come out. So what would happen is the neurotransmitters will go out and into the synaptic cleft, and eventually they can be detected by a receptor. So there are 4 steps or more we think of this way. The first 2 are calcium channels open. They're voltage gated, so the action potential gets to them and they open. And that's the whole purpose of the action potential, is to get to these calcium channels and open them very quickly. They will initiate binding between the 2 snare proteins. Then those snares will allow the vesicle to dock, basically, with the inside of the membrane and fuse. The vesicle and the membrane will fuse, and the neurotransmitters come out. So you could look at this in many different ways, but the key steps are attachment between the V snares and the T snares, and then they bring the vesicle with its membrane into contact with the cell membrane. And when the membranes fuse, then the internal contents of the vesicle get released. Then, ideally, the neurotransmitters cross the synapse, bind with receptors, and ion channels open downstream. And then what would happen is we would think about, where do the neurotransmitters go? How does this process stop? Because if the neurotransmitters have crossed the synaptic cleft, now they're binding to receptors. And eventually they'll fall off of the receptor, and then they'll fall back onto the receptor if they can. That's a tricky thing in chemistry. If you have a ligand in a receptor, and it'll stick together for a while, and then it'll come out, and if it's still there, it'll go back in, and that will keep happening. And we can't let that happen, because that would really slow down our nerve signaling. So what we're going to see is there are ways to get the neurotransmitters back out of the synaptic cleft. That brings us to what we call termination of neurotransmitter. And this is where the nervous system has ways of getting neurotransmitters out of the synaptic cleft. So one thing we have is enzymes that can break down neurotransmitters. So, for example, if the neurotransmitter is acetylcholine, acetylcholine can be broken down by acetylcholinesterase. And so that enzyme can be working here as soon as the neurotransmitters are released. Some of them will hit the, some acetylcholine would hit the receptors and then fall off. Other acetylcholine would hit the enzyme and be broken down. As long as some of it hits the receptors, then the synapse has done its job, but quickly it needs to be broken down so that it doesn't continually activate channels. Another thing that can happen is it can be brought in through pumps and reloaded into synaptic vesicles. And that is something we would expect. That has to happen. And then another thing that can happen is it can diffuse away from the synaptic cleft, either into the blood, into the glial cells. It can be destroyed or just trafficked elsewhere in the body to be reprocessed. So it's either going to be neurotransmitters are either destroyed by enzymes, brought back into the presynaptic neuron, or they diffuse away and are destroyed. So that is termination of the neurotransmitters. And that brings us to the end of this video for physiology of the nervous system, and everything else we talked about in lecture. So that's the end of these videos for now. Thanks.